Hello everyone, I am Aditee Nilkanth Vishe from Shivajirao, Jondhale College of Pharmacy, Asangaon, studying in second year B Pharmacy. Today I am going to explain you the topic from the Medicinal Chemistry first of S.Y.B.Pharm fourth semester, that is Biosynthesis and Catabolism of Catecholamine, under the guidance of Dr. Nitin Moire as a professor and principal of our college. Catecholamine, epinephrine, adrenaline, norepinephrine, noradrenaline, dopamine, they are found within the CNS, PNS and adrenal glands. Introduction. Catecholamines. Neurotransmitters of this group share two chemical similarities, one core structure of catechol and an amine group. Tyrosine is obtained from dietary protein and is transported from the blood into the brain. Each step in the formation of catecholamines depends on a specific enzyme that acts as a catalyst for that step. Tyrosine hydroxylase is the rate-limiting enzyme in the pathway. It determines the overall rate of dopamine and norepinephrine formation. Here is a representation. Phenylalanine get hydrolyzed into tyrosine in liver. Tyrosine get hydrolyzed into dopa by the enzyme tyrosine hydroxylase. In cytoplasm of neurons, dopa convert into dopamine with the help of enzyme dopa decarboxylase, in which CO2 is removed in cytoplasm of neurons. Dopamine first catecholamine form. Dopamine is converted into noradrenaline with the help of enzyme dopamine beta hydroxylase. Norepinephrine form. Noradrenaline get converted into adrenaline with the help of enzyme phenylethanolamine N-methyl transferase PNMT in the adrenal medulla. Catecholamine synthesis, release and inactivation. Tyrosine hydroxylase catalyze the rate-limiting step in catecholamine synthesis. Catecholamine formation can be increased by the administration of a biochemical precursor, example to be converted into a particular neurotransmitter such as L-dopa for the treatment of Parkinson's disease. Catecholamine formation can be decreased by the drug AMPT, alpha-methyl para-tyrosine. This compound blocks tyrosine hydroxylase, thus preventing overall catecholamine synthesis and causing a general depletion of DA and any neurotransmitters. AMPT treatment caused a return of depressive symptoms in patients who had previously recovered following treatment with antidepressants that act selectively on the noradrenergic system, indicating that the depressed patient's recovery depends on the maintenance of adequate levels of catecholamine in the brain. Dopaminergic synapse and dopamine metabolism. First is biosynthesis, second release, third uptake, fourth receptor-mediated signaling, and fifth one is catabolism. Storage and release. Catecholamines are stored in and released from synaptic vesicles. Catecholamines have been synthesized. They are transported into synaptic vesicles for later release. Catecholaminergic neurons use a vesicular monoamine transporter VMAT to transport neurotransmitter molecules from the cytoplasm of the cell to the interior of the synaptic vesicles. Release of catecholamines normally occurs when a nerve impulse enters the terminal and triggers one or more vesicles to release their contents into the synaptic cleft. Psychostimulants such as amphetamine and methamphetamine can cause a release of catecholamines independently of nerve cell firing. Catecholamine depletion, sedation and depressive symptoms. Catecholamine release, behavioral activation. Catabolism of catecholamines. It is the removal of catecholamines and also termination of action of catecholamine. It is in this process structure of catecholamines change with the help of enzymes MAO, COMT, so they do not interact with adrenergic receptor to produce effects. Representation. Enzymes MAO is a monoamine oxidase deamination. COMT is catechol O-methyl transferase methylation in ring. Metabolic breakdown. There are two enzymes mainly involved in the breakdown of catecholamines, catechol O-methyl transferase COMT and monoamine oxidase MOA. In humans, dopamine has only one major metabolite, homovanillic acid HVA. Norepinephrine has two major metabolites, 3-methoxy, 4-hydroxyphenyl glycol. MHPG, and vanillin mandelic acid VMA. Measurement of these metabolites in various fluid compartments, example blood, urine, and cerebrospinal fluid, facilitate in determining the possible involvement of these neurotransmitters in mental disorders such as schizophrenia and depression. MAO inhibitors, the treatment of depression, COMT inhibitors as a supplemental therapy to enhance the effectiveness of L-DOPA in treating Parkinson's disease. I am thankful to our President Dr. Shivajirao S. Jondhale Sir, Secretary Honorable Dr. Geeta Khare Ma'am, the Principal Dr. Nitin More Sir for encouragement and guidance for efficient use of social media to spread knowledge through the making informative videos. Please like and subscribe our channel for such informative videos. Thank you.